Welcome back to the deep dive. Today, we are taking you way beyond the sort of usual suspects like the cell membrane or the nucleus.
We're diving deep into the actual infrastructure of life itself, this incredibly dynamic, always shifting internal network. It's what gives a cell its shape, its movement, really its whole identity, the cytoskeleton. And it's quite a system, isn't it? You know, often when people picture a cell, they think of this almost inert blob, maybe a fluid-filled sac.
Right, just floating around.
Exactly. But the reality, and our source material makes this really clear from the start, is that the cell is more like a meticulously engineered, highly organized city. The cytoskeleton, yes, it's the internal scaffolding, that's true.
The framework.
The framework, but it's also, crucially, the cell's entire transport network. It's like rapid transit system, and even its way of sensing the outside world. It's incredibly complex.
I think that word dynamic is just so key here. We're definitely not talking about, static steel beams holding up a roof.
Not at all.
This internal structure is constantly, and I mean constantly, tearing itself down and rebuilding in response to signals. It's what lets a single cell crawl across a surface. It's what allows your muscle fibers to contract, and it's what keeps all those tiny organelles inside precisely organized, not just rattling around. And those core functions, they're absolutely non-negotiable for the cell to even survive. The sources really hammer home three massive roles. First, maintaining the cell's shape and that intricate internal organization.
Okay.
Second, accomplishing movement. And that's both the whole cell moving, think of like an immune cell chasing down bacteria.
Right, macrophages.
Yeah.
Exactly. Yeah. And also moving stuff inside the cell, like shuttling little packages, vesicles around. And third, something that sounds maybe a bit abstract, but is critical, generating and maintaining cell polarity. Polarity, that comes up again and again.
Yeah.
It's basically what allows cells to specialize, right? To have a top and a bottom.
Precisely. If you're, say, an epithelial cell lining your gut, you absolutely need a distinct top surface, the apical side, to absorb nutrients.
And then you need a distinct bottom surface, the basolateral side, to pass those nutrients out into the bloodstream or tissue. Without the cytoskeleton setting up and maintaining that top-bottom difference, the whole system just it fails.
It's like a loss of identity for the cell. It doesn't know which way is up anymore.
Exactly. And this whole essential system is built from three distinct types or classes of protein filaments. They're really differentiated by their size, what they're made of, and crucially, their overall stability and how dynamic they are. Okay, so let's lay out the three key players for you, the listener. We've got the smallest and probably the most dynamic ones, the actin microfilaments. So there's a middle tier. These are built for serious mechanical resilience. The intermediate filaments, right? And finally, the largest ones, the kind of heavy lifters and long-distance transporters, the microtubules. So our mission today is to really unpack each of these one by one. We want to understand not just the structure, but the specific mechanics, how they actually do their jobs. Sounds like a plan. It's a fascinating world inside there. Okay. All right, let's kick things off with the thinnest and arguably the most abundant system, actin microfilaments. If the cell needs to move fast or reshape its surface or maybe pull something in quickly, it's usually calling on these guys, these fine, dynamic sort of ropes. Yeah, ropes is a good way to think about them. At just 5 to 9 nanometers in diameter, they are definitely the thinnest class. And the basic building block, the monomer, is this globular protein called G-actin.
G for globular.
G for globular, exactly. And it's worth just pausing for a second to appreciate that G-actin is actually the most abundant protein found inside the cytosol of eukaryotic cells.
Wow, the most abundant.
By far. Yeah. Its sheer quantity just tells you how fundamental it is for basic cell function. Everything relies on it in some way.
So these G-actin monomers, they link up, right? They assemble non-covalently, meaning not super strong bonds, into these long rope-like filaments called F-actin. F for filament.
F for filament, you got it.
Right.
And these F-actin filaments then twist around each other to form a double-stranded helix. Yeah. But the real core mechanic here, the thing that makes actin so special, is the filament's inherent polarity.
Okay, polarity again, what does that mean for actin?
Well, it means the filament isn't the same at both ends. It has a structurally distinct plus end and a minus end. Think of it like having a head and a tail. And the plus end is where polymerization, the rapid addition of new G-actin subunits, happens most readily. It's the fast-growing end. The minus end, conversely, is where dissociation or the loss of subunits tends to occur. It's the slower or shrinking end.
So subunits add on one end, fall off the other.
Exactly. And this structural asymmetry is what drives this incredible dynamic behavior we call actin treadmilling.
Treadmill. Okay, that's a great visual, like a treadmill belt, right. The overall structure might look like it's staying in place, but the individual components are constantly moving through it.
Perfect analogy. New subunits hop on the front, the plus end, push everything along, and older subunits fall off the back, the minus end.
So what powers this? It can't just be random sticking and unsticking, right? There must be some energy involved.
Absolutely. You've hit the key regulatory point. The assembly of G-actin needs energy, and it gets it from ATP, adenosine triphosphate.
Yeah.
G-actin binds to an ATP molecule.
The cell's energy currency.
The cell's energy currency. This ATP-bound G-actin is like an energized, high-affinity building block. It really wants to join the filament. Now, once it's incorporated into that growing F-actin filament, the bound ATP doesn't just stay there. It gets slowly hydrolyzed, broken down into ADP adenosine diphosphate plus a phosphate group.
Ah, okay. So the subunits at the growing end, the plus end, are mostly ATP bound. High energy, sticking tightly.
Precisely. They form strong bonds with their neighbors.
Yeah.
But as the filament sort of ages, meaning as subunits get pushed further back towards the minus end, that ATP hydrolysis catches up.
So the older subunits become ADT bound.
Right. An ADP-bound actin has a much lower affinity for its neighbors. It doesn't stick as tightly. This essentially destabilizes the filament structure at that minus end, making it more likely for those ADP actin subunits to fall off.
So you get this gradient. ATP at the front, ADP at the back.
Exactly. This ATP-ADP hydrolysis gradient is the engine. It ensures this constant directional turnover. It allows the filament to push forward, to remodel quickly, which is absolutely vital for things like cell migration, letting the cell crawl.
That makes so much more sense now. It's not just a rope. It's a self-assembling, energy-driven motor element. And speaking of motors, when we talk about actual movement, like muscle contraction or moving cargo, we have to bring in actin's main partner, right? The motor protein myosin.
Myosin is the workhorse. Everyone learns about the really famous, highly coordinated system in our skeletal muscles, right? Where you have these thick filaments of myosin II grabbing onto and sliding the actin filaments past each other.
Yeah, the sliding filament model that
Right.
generates that powerful, rapid, visible contraction.
Exactly, but myosin proteins are actually way more diverse than just the muscle version. In non-muscle cells, you find a whole family of different myosins doing all sorts of jobs.
So how does myosin work in, say, just a regular skin cell or a neuron? What's it doing there?
Well, the diversity allows for specialization. For instance, there's a type called myosin V. It's often involved in high-speed, long-distance transport of vesicles, literally walking organelles along the actin tracts like a delivery truck.
Like walking along the actin rope.
Yeah, step by step, using ATP for energy. Then you have myosin II, the same type found in muscle, but less organized in non-muscle cells. It's often concentrated in the cell cortex, right under the plasma membrane.
Okay. And its contractile ability there allows the cell to do things like pinch itself in two during cell division or to apply tension to its surroundings, maybe pulling on the extracellular matrix. The source specifically mentions these non-muscle roles. Vesicular transport, regulating the tightness of epithelial barriers and driving those less coordinated, broader movements involved in cell migration. Okay? So the actin-myosin system is basically the cell's internal construction crew and its local delivery service. It's all about rapid assembly, rapid breakdown, and just immense responsiveness to signals.
That's a great summary. Dynamic and responsive are the key words for actin.
Okay, so if actin filaments are the dynamic ropes for quick changes, let's move up the size scale. We get to the intermediate filaments. These are about 10 nanometers across. And you describe these as the static, heavy-duty, tensile strength cables. Quite a contrast.
It really is a night and day difference. While actin and, as we'll see, microtubules are defined by their rapid, polarity-driven turnover and remodeling. Intermediate filaments, or IFs, usually don't do that.
They don't actively reorganize much, generally no. Their turnover rates are much, much lower. They also don't have that distinct plus-minus polarity like actin or microtubules. They form these incredibly strong rope-like polymers, and they're really built for one main thing: enduring mechanical integrity, stability. So that stability is directly linked to their function, which you said is providing tensile strength, resisting pulling forces.
Exactly. They are literally the internal structures that prevent a cell from just rupturing when it gets stretched or pulled. Think about cells in your skin. They're constantly being stretched and pulled.
Or like bladder cells as the bladder fills up.
Perfect example. Or cells lining your blood vessels, dealing with the constant pressure and sheer stress of blood flow. EVFs provide the strength to withstand that without the cell tearing apart.
How do they get that strength? What's different about their structure compared to actin?
Their assembly is unique. They don't just form simple linear chains. Monomers first pair up into dimers, then two dimers associate in a staggered antiparallel way to form a tetramer. This tetramer is the basic soluble subunit.
Okay, so dimers make tetramers.
Right, and then these tetramers assemble end-to-end and also pack together side-to-side laterally to build up this thick, complex, interwoven, rope-like filament. It's not a simple helix like actin, it's much more like a multi-stranded rope or cable.
Okay. That structure makes sense for strength and flexibility. It can bend without breaking easily.
Precisely. Gives them incredible resistance to physical force, letting cells bear significant loads without structural failure.
Now, what I found really fascinating in the notes was the heterogeneity here. You mentioned actin and tubulin, which makes microtubules, are basically single, highly conserved proteins across different cell types, but intermediate filaments are different.
Massively different. This is probably the most specialized cytoskeletal component in terms of its protein family. There isn't just one intermediate filament protein. There's a huge, diverse family of IF proteins, and crucially, they show tissue-specific expression patterns.
Meaning different cell types make different kinds of intermediate filaments.
Exactly. And this isn't just some, minor biological detail. It's actually a cornerstone of clinical pathology because, say, a lung epithelial cell expresses one specific type of IF protein and a muscle cell expresses a completely different type.
So pathologists can use that, like a fingerprint.
Precisely like a fingerprint. They use antibodies against these specific IF proteins, it's called immunohistochemistry, to stain tissue samples. If they find a tumor, but it's poorly differentiated, meaning it doesn't look like any normal tissue, they can stain it for different IFs. The type of IF protein it expresses can tell them the likely cell of origin. It's like cellular forensics.
That's incredible. Can we walk through some of the key examples the sources mentioned, starting with epithelial cells like skin or gut lining?
Sure. Epithelial cells, which form all our linings and glands, rely heavily on a class of IFs called keratin proteins, or sometimes cytokeratins.
Keratin, like in hair and nails.
Related, yes, but these are the internal cellular keratins. There are actually over 30 distinct types known in humans, often expressed in pairs depending on the specific epithelial layer. And their structural importance is just immense. We know this because mutations in specific keratin genes cause these devastating blistering diseases, a group called epidermolysis bullosa.
And what happens there?
Basically, if the keratin IF network connecting the skin cells together or anchoring them to the underlying tissue is faulty, the cells literally cannot withstand mechanical stress. Even minimal friction causes the cells to rupture, leading to severe blistering and fragility of the skin.
Wow. So a failure in this static structural component has immediate catastrophic functional consequences.
Absolutely. Structure is function here.
Okay, what about connective tissues, like fibroblasts that make collagen or endothelial cells lining blood vessels?
For those types of cells, generally classified as mesenchymal cells, the main intermediate filament protein is the mentin.
Vimentin, okay.
So if a pathologist finds a tumor somewhere else in the body, say a metastasis, and it stains positive for vimentin but negative for keratins, they immediately know it likely didn't start in an epithelium. It probably originated from a connective tissue source, like a sarcoma from fibrous tissue, or maybe a melanoma, which derives from neural crest cells that express vimentin. It dramatically narrows down the possibilities.
Super useful diagnostically. And what about highly specialized cells like muscle or nerve cells?
They have their own specific IFs, too. In muscle cells, the key one is desmin. Desmin forms this critical scaffold around the actual contractile machinery, the actin and myosin bundles, sarcomeres.
So it doesn't make the muscle contract, but it organizes the parts that do.
Exactly. It links the sarcomeres together and connects them to the cell membrane. It ensures that the force generated by all those tiny actin-myosin interactions is actually transmitted effectively across the entire muscle fiber and ultimately to the tendon. Without desmin, the muscle structure falls apart under stress.
Okay. And neurons, they have those incredibly long axons.
Right, think about the nerve fibers running down your leg that can be a meter long. They need serious structural support. Neurons rely on neurofilaments. These are specific IFs that are essential for maintaining the structural integrity and, importantly, the diameter and rigidity of these long axons. They resist stretching and prevent the axon from collapsing or breaking.
So IFs are providing strength everywhere, from skin to muscle to nerves. And then there was one more location that was kind of surprising. Inside the nucleus.
Yes, the lamins. These are a unique class of intermediate filament proteins. They don't form networks out on the cytoplasm. Instead, they assemble right underneath the inner membrane of the nuclear envelope. They form a meshwork called the nuclear lamina.
What does the nuclear lamina do?
Well, primarily, it defines the shape and provides mechanical stiffness to the nucleus itself, protecting the genome inside. But it's turning out to be much more than just structural. The lamina interacts directly with chromatin, the DNA, and its associated proteins, and seems to play a big role in organizing the genome and even regulating gene expression, turning genes on or off.
So the cell's physical architecture, right down to the nucleus, is actually influencing its genetic activity. That's mind-blowing.
It really connects structure and function at the deepest level. And strikingly, mutations in lamin genes are linked to a range of diseases called laminopathies, including some forms of muscular dystrophy, and perhaps most dramatically, premature aging syndromes like Hutchinson-Gilford progeria syndrome.
Progeria, the rapid aging disease, that's
Yep.
linked to nuclear intermediate filaments.
Yes, it highlights how fundamental this structural scaffolding is, not just for cell integrity, but somehow for the whole organism's developmental timing and lifespan. Failure here has profound consequences.
I
Okay, moving on to the third and final pillar, the largest component, microtubules. These are the big ones, 25 nanometers across. These hollow tubes. I always kind of picture them as the cell's major railway lines or highways, really organizing long-distance transport. That's a perfect analogy. They're definitely the cell's high-speed, long-distance transit system. Microtubules are built from building blocks that are actually dimers, pairs of two related proteins, alpha-tubulin and beta-tubulin. These dimers polymerize non-covalently end-to-end to form a protofilament, and typically 13 of these protofilaments associate side by side to form the wall of the hollow tube.
So alpha and beta tubulin dimers make the tube. And you said earlier they're also dynamic and polarized, like actin.
Yes, highly dynamic and highly polarized, but their dynamics are often sort of operating on a grander scale in actin. They really govern the overall spatial organization of the entire cell. And like actin, they have a distinct plus end and minus end, which dictates the direction of growth and shrinkage.
But unlike actin, which seems to be able to pop up almost anywhere it's needed, microtubules usually grow from a specific spot, right? A central hub.
That's a key difference. In most animal cells, microtubules originate from a primary microtubule organizing center, or MTOC. This is usually located near the nucleus and contains the centrosome, which itself contains a pair of barrel-shaped structures called centrioles.
The centrosome is the
The
MTOC.
centrosome is the major MTOC in animal cells, yes. And the reason it acts as the hub is because it contains these specialized ring-shaped structures made of another type of tubulin called gamma-tubulin. These gamma-tubulin ring complexes act as nucleation sites templates for new microtubules to start growing.
Ah, so they provide the starting point. And which end of the microtubule is anchored there?
The minus end. The minus end is typically embedded in and stabilized by the MTOC. This means the microtubules radiate outwards from the centrosome, like spokes on a wheel, or maybe like searchlight beams.
Okay, so the MTOC anchors the minus end near the center, and the microtubules grow outwards with the plus end doing all the exploring out towards the cell edge.
Exactly. And that plus end is incredibly dynamic. The process governing its behavior is called dynamic instability. It's not quite like actin treadmilling. It's characterized by these relatively long periods of steady growth called rescue, which can be suddenly interrupted by rapid catastrophic shrinkage called catastrophe.
Catastrophe. Sounds dramatic.
It can be. The filament can just rapidly depolymerize, shrinking back significantly. Then it might get rescued and start growing again. The plus end is constantly elongating or receding, adding or subtracting tubulin dimers, primarily in response to cellular signals and needs. This allows the cell to rapidly rearrange its entire internal highway system or to probe a new direction if it's migrating.
And it's these microtubule tracks, these highways, that provide the framework for those molecular motor proteins you mentioned earlier.
Precisely. They serve as the essential tracks or mooring lines for specific motor proteins. We have two major families of these motors that literally walk along the microtubule, using the energy from ATP hydrolysis to take steps, and they carry cargo vesicles, mitochondria, even clusters of proteins or mRNA molecules throughout the cell.
Okay, so these are the engines for the railway system, and you mentioned two main families.
The two major players are the kinsins and the dyneins. They are like the traffic controllers, ensuring cargo moves in the correct direction along these polarized tracks.
How do they know which way to go?
They are structurally built for directionality. Kinisins, as a general rule, typically walk towards the plus end of the microtubule.
So away from the cell center, the MTOC, towards the periphery, the outbound traffic.
Exactly. Kinisins handle most of the anterograde transport, moving things out towards the cell surface or the end of an axon, for instance. They're the outbound delivery trucks.
Okay, so Keenans go out. Then Dynins must handle the return trip.
Yes, generally speaking, dyneins are primarily responsible for retrograde transport, moving cargo back towards the minus end, towards the MTOC and the cell center. So they bring materials back from the periphery, maybe for recycling or processing, near the nucleus.
And the specificity of these motors, kinesin for outward, dynin for inward, is what ensures organelles end up in the right places.
It's absolutely critical, especially in very large, polarized cells like neurons. Think about getting newly made proteins or mitochondria all the way down that meter-long axon. That relies entirely on efficient kinase-driven transport. And bringing back old components or signaling molecules from the synapse relies on dynein. If this microtubule-based transport system fails in neurons, it leads directly to severe neurodegenerative diseases. The connections just break down.
So transport is a huge job. But microtubules also build some really complex, large-scale structures, right? Especially for things like cell division and motility.
Yes, beyond just being tracks, they have these major architectural roles. The source highlights two really crucial ones. First, mitosis, or cell division.
Ah, the spindle fibers.
Exactly. The mitotic spindle, that complex machine that segregates the duplicated chromosomes, is built almost entirely of microtubules. They radiate out from the duplicated centrosomes, which become the spindle poles, capture the chromosomes at structures called kinetotors, and then precisely pull the sister chromatids apart to opposite ends of the cell. The entire fidelity of cell division hinges on the precise, highly regulated dynamic instability of these spindle microtubules.
So errors in microtubule function during mitosis would be catastrophic for genetic inheritance.
Absolutely. It leads to aneuploidy, the wrong number of chromosomes, which is often lethal or a hallmark of cancer cells.
Okay, mitosis is one major role. What was the second one? Specialized projections.
Right, things like cilia and flagella. Microtubules form the structural core, the axoneme, of these cellular appendages.
Like the flagellum on a sperm cell for swimming? Or the cilia in our airways that move mucus?
Exactly. Those are motile cilia and flagella. They have a very specific, highly conserved internal arrangement of microtubules, typically described as the 9+2 structure. 9 doublet microtubules, arranged in a ring around 2 central single microtubules. Coordinated sliding between these doublets, driven by dynein motors, generates the bending motion that allows swimming or sweeping.
Okay, the 9+2 for movement. But aren't there other kinds of cilia too?
Yes, and this is incredibly important. Most cells in our body actually have a single non-motile cilium called a primary cilium. These typically have a 9+0 structure, lacking the central pair and associated motile machinery. So if they don't move, what do primary cilia do?
They function as crucial cellular antennae. They're covered in receptors and signaling molecules, sensing chemical and mechanical signals from the cell's environment. They play vital roles in development, tissue maintenance, and regulating cell proliferation and differentiation. And critically, we now know that mutations affecting the proteins that build or function within primary cilia cause a wide range of human diseases known collectively as ciliopathies. Like what kind of diseases?
Polycystic kidney disease is a major one. Also, certain forms of retinal degeneration, skeletal abnormalities, and even obesity and developmental disorders. It turns out these little antennas are coordinating a huge amount of cellular activity.
Wow. So the architecture built by this tubulin system isn't just about moving cargo internally. It's fundamental for cell division, for movement, and even for sensing the outside world and coordinating development. They're really central players.
Absolutely central. From internal highways to external sensors and reproductive machinery.
Okay, so we've looked at the three pillars individually: the quick, flexible actin, the stable, strong, intermediate filaments, and the robust microtubule highways. Now let's talk integration, because, structure is kind of useless if it's just isolated components floating around. How does this internal skeleton actually work together to define cell identity, especially that polarity concept we keep mentioning, and how does it physically connect the cell to its neighbors and the outside world?
Right, integration is absolutely key. It's where these systems really come together to create a functional whole. And it definitely circles back strongly to polarity. The cytoskeleton doesn't just prop the cell up. It actively maintains that asymmetrical organization that's essential for specialized function, particularly in tissues like epithelia.
So it's the arrangement of these networks that keeps the top distinct from the bottom.
Exactly. The strategic placement and anchoring of specific microtubule networks, often oriented along the apical basal axis, and the distinct actin networks found, for instance, just under the apical membrane, forming the terminal web, ensure that the apical surface maintains its unique protein and lipid composition, separate from the basolateral surface.
And that separation is what allows for that vectorial transport you mentioned, moving things directionally across the cell.
Precisely. It allows an intestinal cell to absorb glucose only at the top surface and export it only out the bottom surface towards the bloodstream, or for a kidney tubule cell to reabsorb salt from one side and pump it out the other. It's fundamental to organ function.
Okay, so the internal arrangement dictates polarity. But how is this physically enforced? How does the cell rivet itself in place and connect to others? Through junctions, right?
Yes, through specialized anchoring junctions. These are protein complexes that physically link the cytoskeleton inside the cell across the membrane to either adjacent cells or to the non-cellular scaffold outside the cell, the extracellular matrix or ECM. And importantly, these aren't just passive rivets, they're major signaling hubs too.
Right, structure and signaling combined. Let's maybe break down the main types of anchoring junctions based on which cytoskeletal element they connect to inside the cell, starting with the ones linked to actin.
Okay. The primary cell-to-cell junctions that link to the actin cytoskeleton are the adherence junctions. They use transmembrane proteins from the cadherin family, primarily E-cadherin in epithelia, to stick cells together.
Cadherins bridge the gap between cells.
Right. And then inside each cell, the cytoplasmic tails of these cadherins are linked via a complex of adapter proteins like catenins directly to the actin microfilaments.
Ah, so they connect to the most dynamic system. What does that imply?
It implies that these junctions are also potentially quite dynamic. The connection to actin allows these junctions to influence and be influenced by changes in cell shape, tension, and motility. They help form the adhesion belt, often seen near the apical surface of epithelial cells, which can contract, using associated myosin, to help shape tissues during development, for example.
And this is where that link to cancer pathology becomes really clear, right? With E-cadherin.
It's a classic textbook example. The loss of functional E-cadherin, either through mutation or just shutting down its expression, disrupts this crucial link between adjacent cells and their internal actin cytoskeletons.
So the cells stop sticking together properly.
Exactly. They become discohesive. They lose that strong cell-to-cell adhesion. This change is a hallmark of a process called the epithelial to mesenchymal transition, or EMT, where organized epithelial cells transform into migratory, invasive, mesenchymal-like cells. This loss of E-cadherin-mediated adhesion is seen very commonly in certain types of invasive cancers, like infiltrative lobular breast cancer or diffuse gastric cancer. It structurally enables them to break away and metastasize.
Well, literally, the breakdown of this specific structural link to the actin system can unleash invasive cancer.
It's a major contributing factor, yes. Structure dictates behavior.
Okay, so adherens junctions link to actin for dynamic cell adhesion. What about the junctions that need serious strength, the ones linked to intermediate filaments?
Those are primarily the desmosomes for cell-to-cell adhesion and hemidesmosomes for cell to extracellular matrix adhesion. These are the heavy-duty anchors.
Let's take desmosomes first. They also link cells together.
Yes. Desmosomes also use cadherin family proteins, specifically desmogleins and desmocollins, to bridge the gap between adjacent cells. But internally, instead of linking to actin, they connect via adapter proteins, like plakoglobin and chalakin, to the intermediate filaments, typically keratin filaments in epithelial cells.
Ah, so they anchor to the cell's strongest tensile cables.
Precisely. Because the intermediate filament network is so strong and extends throughout the cytoplasm, the desmosome effectively distributes mechanical stress pulling, shearing forces across the entire sheet of cells. It prevents the tissue from tearing apart under load, like in the skin or heart muscle.
Okay, that makes sense for tissue integrity. Now, hemidesmosomes, the hemis, suggest they're only half a desmosome, connecting to something else.
Exactly. Hemidesmosomes anchor the cell not to another cell, but to the underlying extracellular matrix, ECM, specifically the basement membrane or basal lamina that epithelial cells sit on.
The foundation.
The foundation. And the transmembrane connector proteins here aren't cadherins. They are primarily from the integrin family. These integrins span the membrane, binding to ECM components like laminin outside the cell. And inside the cell, they link, again via adapter proteins, to the intermediate filaments, usually keratins.
So integrins link IFs to the ECM. It's the cell's main anchor point to its substrate.
It's a crucial anchor point, yes, providing stable adhesion, especially important in tissues subjected to shear forces, like skin or the lining of the gut. And this brings us, I think, to that really complex integration point you hinted at earlier, the focal adhesion complexes. This is where the cell isn't just anchored, but it starts listening to the forces it feels through those anchors. You've nailed it. Focal adhesions are related to hemidesmosomes in that they also involve integrins linking the cell to the ECM. However, focal adhesions typically link the integrins inside the cell to the actin cytoskeleton, not intermediate filaments. But the principle of mechanosensing applies strongly to both types of integrin-mediated adhesions. These sites where integrins cluster are much more than just anchors. They are dynamic signaling platforms.
Okay, wait. So focal adhesions link integrins to actin, and hemidesmosomes link integrins to intermediate filaments.
That's the general distinction, yes. Focal adhesions are often more associated with cell migration and sensing the stiffness of the substrate via the dynamic actin network. Hemidesmosomes provide that superstable anchoring via the IF network. But both use integrins as the transmembrane link to the ECM, and both are sites where mechanical force can be translated into intracellular signals.
Let's focus on that translation process. How does a physical pull on an integrin molecule get turned into a chemical signal inside the cell? This is mechanobiology, right?
Exactly. This is the core of mechanobiology. When the ECM pulls on an integrin, or the cell pulls on the ECM via its cytoskeleton, the integrin molecule literally changes its shape, its conformation.
Okay. This conformational change does two things. It strengthens the integrin's binding to the ECM and the cytoskeleton, and critically, it causes signaling proteins that are clustered around the integrin's cytoplasmic tail to become activated. What kind of signaling proteins hang out there?
A whole host of them. Key players include protein kinases, enzymes that add phosphate groups to other proteins. Two really important ones found in these adhesion complexes are focal adhesion kinase, FAK, and CYTARC kinase. When the integrin gets pulled and changes shape, FAK gets activated, often by autophosphorylation, and it then phosphorylates other proteins, including CYTARC, kicking off a whole cascade of downstream signals.
So the tug activates FAK and CYTARC, and they start sending messages deeper into the cell.
That's the essence of it. The message might be, we are adhered, or we are under tension, or this surface is stiff, or this surface is soft. And the cell then responds appropriately, maybe by strengthening its adhesion, changing its shape, migrating, proliferating, differentiating, or even undergoing programmed cell death, apoptosis, if the signals are wrong. It's how cells read the physical nature of their environment.
And this is happening at both hemidesmosomes, linking IFs, and focal adhesions, linking actin.
The principle of integrin-mediated mechanotransduction happens at both, yes. The specific downstream signals and cellular responses might differ, depending on whether the connection is primarily to the stable IF network or the dynamic actin network. But the fundamental idea of converting mechanical force into biochemical signals via integrins is conserved.
Okay, this is fascinating. We've got the components, the dynamics, the integration, the signaling. Let's pivot now towards the cutting edge. We know how these systems work, roughly, but what are some of the really important implications and applications, especially when things go wrong in disease, or how we might exploit this knowledge in medicine?
Well, probably the most direct and well-established application, building right on the dynamics we discussed, is in cancer therapy. Specifically, targeting the microtubule system.
Because microtubules are essential for mitosis, for cell division.
Absolutely essential. We talk about how the dynamic instability, the rapid growth and shrinkage of spindle microtubules is absolutely required to capture chromosomes and pull them apart correctly.
Right, so if you mess with that dynamism,
you can stop cell division in its tracks. And since cancer is fundamentally a disease of uncontrolled cell proliferation, microtubules became a prime target for chemotherapy very early on.
So how do these chemo drugs work? Do they break the microtubules?
They work in two main ways, targeting different aspects of the dynamics. One major class are the microtubule destabilizers. These include drugs derived from the periwinkle plant, the vinca alkaloids like vinblastine and vincristine.
Okay, destabilizers, what do they do?
They basically bind to free tubulin dimers and prevent them from adding on to the microtubule plus ends. They inhibit polymerization. This leads to a net depolymerization, causing the mitotic spindle to collapse, arresting the cell in mitosis, which usually triggers apoptosis.
So they stop the building process. Is there an opposite approach?
Yes, there is. The other major class are the microtubule stabilizers. The most famous examples here are the taxanes, like paclitaxel and Taxol, originally derived from the bark of the Pacific yew tree.
Stabilizers. How does stabilizing them stop cell division?
It seems counterintuitive, but it works just as well, if not better sometimes. Taxanes bind directly to the microtubules themselves, not the free dimers, and they suppress dynamic instability. They lock the microtubule in a polymerized state, preventing it from shrinking or depolymerizing when it needs to during mitosis.
Ah, so the spindle forms maybe captures chromosomes, but then it can't shorten or adjust properly to pull the chromosomes apart.
Exactly. The cell gets stuck with this hyperstable, non-functional spindle. Again, this leads to mitotic arrest and cell death. So you can kill cancer cells either by preventing microtubule assembly or by preventing disassembly. It highlights just how critical that precise regulation of dynamic instability is.
That's a really clear example of exploiting fundamental cell biology. What about polarity again? We talked about its importance. How does its loss manifest in disease beyond the E-caterin example in cancer?
Well, the loss of that clear apical basolateral distinction in epithelial tissues is often an early warning sign in many diseases, including chronic inflammatory conditions and the development of precancerous lesions, dysplasia. When the tight junctions, which seal the gap between cells apically, and the underlying cytoskeletal organizations start to break down.
The vectorial transport fails.
The directional transport collapses, yes. But also, cells might lose their proper orientation. They might start piling up on each other inappropriately. The tissue architecture degrades. This can impair organ function long before you see overt cancer. Think about kidney tubules losing their ability to properly filter and reabsorb, or glands losing their specialized secretory function. Monitoring tissue polarity is becoming increasingly important diagnostically.
Okay. And let's revisit the intermediate filaments as diagnostic tools. You mentioned how pathologists use vimentin versus cytokeratins to determine the origin of metastases. Can you just re-emphasize how valuable that simple test is?
Oh, it's incredibly valuable. It can literally save weeks of diagnostic uncertainty and immediately guide the oncologist towards the most appropriate treatment strategy.
Yeah.
Imagine a patient presents with spots in their lungs on a CT scan metastatic disease. The primary tumor could be almost anywhere.
Right. Lung, colon, breast, kidney, melanoma, sarcoma, endless possibilities.
Exactly. The pathologist takes a biopsy of the lung lesion. They stain it with antibodies. If it lights up brightly for cytokeratins, they know immediately there's a carcinoma, an epithelial cancer. They can then use more specific cytokeratin markers, like CK7 and CK20 patterns, to further narrow it down. Is it likely from the lung itself, the colon, the breast, etc.?
Okay. Conversely, if it stains strongly for vimentin and is negative for keratins, they know it's almost certainly not a carcinoma. It's likely mesenchymal in origin, maybe a sarcoma that started in bone or soft tissue, or a melanoma. This points towards completely different treatment approaches. It's a relatively quick, robust way to get a fundamental piece of information about the tumor's identity just by reading its structural signature. It really is like reading the cell's fixed address from its internal scaffolding.
That's a perfect analogy.
Okay, finally, let's loop back to mechanosensing, integrins, and those focal adhesions, or hemidesmosomes. You said this field, mechanobiology, is booming. What are the big implications there, especially for common diseases?
The implications are huge, particularly in cardiovascular disease, fibrosis, scarring in various organs, and also osteoporosis and even cancer progression. We're realizing more and more that cells don't just respond to chemical signals, they respond profoundly to the physical properties of their environment.
Like how stiff or soft the surrounding tissue is.
Exactly. For example, the stiffness of the extracellular matrix can influence how stem cells differentiate, whether they become bone cells, fat cells, or nerve cells. In disease, tissue stiffening, like the fibrosis that occurs in the heart after a heart attack or in the liver with cirrhosis, creates abnormal mechanical cues for the cells living there.
And the cells sense this stiffness through their integrin connections to the cytoskeleton.
Yes. Those integrins, linked to either actin or IFs, feel that increased tension or stiffness. This activates those signaling pathways involving FAK and SRC and many others. In the context of heart failure, for instance, the cardiac myocytes, muscle cells, are constantly being overstretched. This chronic mechanical stress, transduced across integrins, leads to persistent pathological signaling cascades inside the cell that drive hypertrophy, cell enlargement, fibrosis, and ultimately contribute to the heart failing.
So the physical strain directly triggers chemical pathways that make the disease worse.
Precisely. It's a vicious cycle where altered mechanics drive pathological signaling. Similarly, in blood vessels, the shear stress from blood flow is sensed by endothelial cells via integrins and other mechanosensors. Abnormal flow patterns can lead to signaling that promotes inflammation and atherosclerosis, plaque buildup.
So understanding this link opens up new therapeutic avenues, trying to interrupt that mechanosignaling.
Absolutely. There's intense research into developing drugs that target FAK or other key nodes in these mechanotransduction pathways. The idea is maybe we can't easily reverse the tissue stiffness itself, but perhaps we can block the harmful signals that arise from it. Other approaches are looking at modulating the integrant ECM interactions themselves, or even using biomaterials with specific mechanical properties to guide tissue regeneration in a more healthy way. It's all about recognizing that mechanics are just as important as chemistry and biology.
Wow, that was genuinely a deep dive into the cell's internal world. Amazing complexity. For everyone listening, let's try to quickly consolidate the main takeaways. Maybe just revisit the key roles of our three main players in simple terms.
Sure. Let's think about their core properties. First, actin microfilaments. Think. Dynamic ropes. They're the thinnest, built from G-actin, highly polarized and constantly remodeling via treadmilling, powered by ATP. Their job is rapid shape change, movement, and generating force, especially with their partner, myosin. Flexibility and response. Okay, dynamic ropes, got it. Second, intermediate filaments. Think. Static cables. They're mid-sized, built from diverse, tissue-specific proteins like keratin or vimentin. They are not typically dynamic or polarized. Their main role is providing tensile strength, resisting mechanical stress, ensuring tissue integrity. They're the resilience providers and great diagnostic markers. Static cables for strength. Okay, and the last one.
Railway tracks, the largest hollow tubes built from alpha-beta tubulin dimers. Highly polarized, plus-minus ends, and exhibit dynamic instability, usually growing from a central MTOC, centrosome. They are the main tracks for long-distance organelle transport via kinesin, outward, and dynein inward, motors. They also build large-scale structures like the mitotic spindle for cell division and the core of cilia and flagella for movement and sensing, organization, and transport.
Dynamic ropes, static cables, and railway tracks. That helps simplify it. So ultimately, the cytoskeleton isn't just some passive internal scaffolding. It's really the cell's engine for transport, its communication system for physical forces, and the absolute guarantor of its identity, its polarity, its place in the larger tissue.
Couldn't have said it better myself. That idea of place brings us right back to where we started, thinking about E. cadherin and cancer. We learned that losing those key cytoskeletal anchor points the links between cells, or to the ECM, is what could allow a cancer cell to break free, to invade, to metastasize. It loses its connection to its neighborhood, essentially.
Right. So here's a final thought to leave you with, building on that. If the cytoskeleton and its connections are truly what define a cell's self and its place within the body's architecture, what if we shifted our thinking slightly? Instead of just trying to poison cancer cells by targeting their dynamic processes like mitosis, what if future research focused more on finding ways to mechanically stabilize those crucial linkages that keep cells in their proper place? Could we perhaps reinforce the E-cadherin-actin connections or strengthen the intermediate filament anchors in epithelial cells, specifically to physically prevent them from detaching and initiating that whole metastatic cascade in the first place?
That's a fascinating angle. Could the most potent anti-metastatic strategy of the future be less about killing the cell and more about simply reinforcing its connections, reminding it structurally where it belongs? It shifts the therapeutic focus from cell dynamics towards tissue mechanics and stability.
Something to definitely think about. Could we glue tumors in place? A provocative idea. That was our deep dive into the cell's dynamic scaffolding. Thanks for joining us.
Thank you.
We'll see you next time on The Deep Dive.